Hello, everyone. Welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld. You have your name. I have my name on yours and yours on mine.
I don't know. I can't see that. I don't even see the questions. So it's not like the split screen that it usually is.
And I can't move mine. Mine is like stuck. I'm just going to take it away. We'll just have your name there.
Okay.
So Amy and I were chatting in the pre-show, and I decided, and folks, you tell me if this is correct, that Amy is the John McEnroe of virology.
Johnny Mac.
Johnny Mac of virology. What do you think of that, folks?
Johnny Mac. Hey, he's really smart. You know, he went to Stanford. He and his brother went to Stanford. They didn't finish, but they went to Stanford.
So they got in?
Yeah. They're really smart. And he's an art. You know, he used to own galleries and play the guitar. He's cool.
So Amy has a purple shirt and a tan sweater. Momo got the cardigan with the button-up shirt, but nobody got the sweater color.
What cardigan? I'm wearing a V-neck.
Isn't that a cardigan?
No. Do you see any buttons?
What's a cardigan? A cardigan is a button-up thing? Well, what do I know? I'm sorry.
Yeah, this is why I dress you.
Actually, someone says you're the Muhammad Ali of virology.
That's interesting. That's fun.
Well, John McEnroe is contentious, right?
Yeah.
And Amy can be contentious.
Blunt. He's just blunt. He just tells you when the guy is sucking it up on the court. He's
and
like,
Amy can be blunt.
Legs, he needs to get it together.
But I have to say, folks, Amy may be blunt, but she's usually right. And so let's start this evening. Amy, where was Paul Offit this week, and what did he say?
Oh, so he was on Andy Slavitt this morning.
And what did he say?
So he said testing is a mess. Flat out, testing is a mess. And that if you are asymptomatic and vaccinated, no testing.
There you go. Has Amy been saying this for a couple of weeks? And how many of you have been giving her a hard time? So
total shit.
Now Offit comes around. It may be that he listened to Amy on Twiv, or on this live stream. So there you go.
He said a few other things that were interesting too.
What else?
So only 15% of children 5 to 12 are vaccinated. So 85% of all kids are not vaccinated.
That's a lot.
It's a huge. You think?
They do think, yeah.
Yeah, it's a huge amount. And then he said, so they don't. So the 3 to 5 year olds were in the trial did not make antibodies. Really, they did not have high titers.
Wait a minute.
So they're thinking.
Wasn't someone arguing with you about that on Twiv recently?
Yeah. And they think that this is automatically going to be fixed with a third booster. And that he had thought that they would have worked it all out before in the phase 1. But he doesn't expect that this is going to go through until the late spring, early summer, somewhere around May. And then I listened to testing also, and that person, didn't bring up the fact that he had originally said lick a stick for under $1. Now he's talking about $4 tests, and I'm like,
why $4? Oh,
I don't know.
They're supposed to be free.
Should be free.
It should be
free.
So
that's what. Yeah. So Offit said how his sister lives in England for the last 30 years, and she visited Offit last week. And in her little travel kit, she brought all the free tests that Health Britain or Health England, whatever it's called, had given her for free.
Mm-hmm.
So if you want to rob a house, look up Paul Offit address, and he has free tests.
Okay. Anyway, just remember, folks, Amy's been talking about not testing asymptomatic people. Many of you said you're
From
wrong.
vaccinated asymptomatic.
Vaccinated, and now Paul Offit endorses that. So, as I've told you many times, Amy is often right, and I should know because I've been working with her for 20 years, and she never lets me forget it. Okay. Now, the other thing I wanted to tell you about very briefly before we start answering your questions, what I think is perhaps one of the most important papers of the pandemic. It's a preprint, so it's not a paper yet. I just want to show it to you, and we can chat about it a bit. And so here it is. I mean, I should make it a little bigger, eh? How can I do that?
I can't see it.
It's on the, it's going to come up. Yeah, there it is on the stream. I can zoom in a bit here. There we go. Infectious viral load in unvaccinated and vaccinated patients infected with SARS-CoV-2 wild type, Delta, and Omicron. Okay. I happen to know Laurent Kaiser here. Remember him? He's from Geneva. He's worked on rhinoviruses with Caroline Taparel.
Oh, yeah. Okay, yeah.
Okay.
Yeah, I know.
So I'm going to just read you the summary here. So what they did is they took nasopharyngeal swabs of patients and they measured RNA by focus forming assay, which is almost as good as a plaque assay, and compared that to RNA genome copies. And they did this during the first 5 days in 384 patients. And so, you know, Amy's always saying one time point is not enough, and she's right. And they did it here. And here's what they found. Excuse me. Correlation between RNA copy number and infectious viral titer was low for all groups. Shocking. Who's been saying use infectivity if you want to study the biology of this virus? Amy has for 2 years now.
Oh, no reason to focus on the details.
No correlation between infectious viral titers and age or sex was seen. We observed higher genome copies in the pre-variant SARS-CoV-2 compared to Delta. Higher genome copies in pre-variant compared to Delta. So don't pay attention to RNA loads. But significantly higher infectious viral titers in Delta-infected individuals. So that could explain the fitness advantage of Delta. In vaccinated versus unvaccinated Delta-infected individuals, RNA genome copies were comparable, but vaccinated individuals have significantly lower infectious viral titers and cleared virus faster. And finally, vaccinated individuals who were infected with Omicron had comparable infectious viral titers to Delta. Oh my gosh. So infectious titers, if you want an answer to the question, look at the right experiment, do infectivity.
Oh, why focus on the detail?
Now, let me show you a couple of sentences, and then we'll stop here. In the I was sending these to Amy and Daniel Griffin tonight while I was riding the train here. This is the first...
In case I hadn't read the paper, right? That was in case I hadn't read the paper. Me.
No, it was mostly for Daniel. It was mostly for Daniel.
Oh, okay.
This is the first study which quantified infectious viral loads in patients infected with different variants. So how many years are we into the pandemic? And this is the first study. We've been calling for this for 2 years now. Okay? Significant reduction of infectious viral loads in vaccinated patients. No difference between Delta and Omicron. This should start to... I wish the press gets this. I'm not sure that they're going to get it. In most studies, viral load is measured as RNA genome copy and not actual infectious virus. In this study, we could show that detection of RNA genome copies in upper respiratory tract swab samples is only poorly correlated with infectious viral shedding. Oh my gosh, this is all Amy channeled. I can't believe it. Finally, one more. With only a qualitative result. So you take a nasal swab and you throw it in a dish of cells and you say, does virus grow or not? That's qualitative. It's not quantitative. Overall virus isolation success cannot distinguish between high and low infectious loads in a sample, a key determinant of the size of the transmitted inoculum. Do you remember, Amy, a study out of a certain Midwestern state where they just threw it onto cells and say, aha, there's infectivity here? Well, that's
I remember because it's the same guy who sent me serum. And then when I said that there was no virus and it said, oh, it's because it's serum and not plasma. And I said, what?
And one more thing.
Remember that?
I do remember, of course. One more thing, and then we'll move on, because we're going to do this paper on TWIV. Look at this. Where is it? I think it's on the next page. I think, yeah. Whether lower infectious viral load translates into lower secondary attack rates remains controversial and depends on other factors like stability of the virus particle. And I just want to say here, while the viral load is a key element of transmission, the process of human-to-human transmission is complex, and other factors, such as recommended protection measures in a certain country at a certain time, overall incidence, perceived risks, the context of contacts, can influence outcomes. Do you remember Amy's New York Times op-ed? This is exactly what it said. Okay, so tonight is Amy was right night, okay? And stop bugging her. She's brilliant, and she's been right on everything she has said. So when she says something, you don't have to take it the first week. Sometimes it takes me weeks, and then I finally come around and I say, damn it, you're right.
Weeks?
You mean months?
Weeks?
Years?
Well, okay, we'll give the average of months. There have been certain things that I've complained about for years that you've come around to, like the lack of time zero on pathogenic graphs and stuff. But there are some other things you get right away, like my antigen stuff.
Okay.
Although you weren't that excited about my last result as I was. I got
podcasting.
That's good, but I was really excited about my last result. I was like, wow.
All right, so let's do some questions.
Oh, okay.
My name is Racaniello, okay? Racaniello. Where can I find the figures that tell the chances of getting severe COVID, triple vax compared to un? Is that the Ontario study, Amy, that she's thinking about?
Yeah.
All right, so that was on a recent TWIV, which you're going to have to go back.
It was about a month ago.
Maybe someone will put it up in the chat as we're going along here. Okie dokie. It's how and what antivirals stamp out this in future pandemics. Thoughts, Amy?
Well, we don't have an antiviral. We haven't really seen it work like in real time, right? It's only been in trials and stuff. So like molnupiravir or whatever the Pfizer drug is, yeah, right. And since those are against the active sites of enzymes and they're conserved basically throughout the entire viral family, I would assume that they're going to be used to quell the next corona pandemic. But I haven't seen it where it's in real time, Could be different in real time than it is in a trial.
Yeah, we'll see. The data should start pouring in. We'll start to get some effectiveness.
Well, are the prescriptions being filled?
Yeah. Animal Party, hello. It says you would rock burlap. Always brilliant. We just love her. That's why we're interested. So you'd look good in burlap, man.
Yeah, burlap's not so comfortable. It's too itchy.
Thank you, Roger. Yes, I appreciate it. I think it's better than the media, but it's not. We don't do it every day, so it's different.
Well, the media has one job. To do it every day. I have 5 jobs. I mean, seriously.
I heard Amy say the vaccines are working well with Omicron. The CEO of Pfizer said he wants 3 separate vaccines just for Omicron, and they will be ready by March. The CEO of Pfizer knows nothing about viruses, okay?
He's a veterinarian. He's a veterinarian.
I don't think you should listen to him. I'm not a fan of Pfizer as a company, but, they did make a vaccine for us, so that then I had it in one of my arms, right arm, I think, yeah. No, you don't need separate vaccines right now. You certainly don't need an Omicron vaccine because everything is looking like the vaccines are working quite well. Amy sent me a slew of papers today. Cross-reactive T cells.
Yeah.
Cross-reactive T cells.
All about the T cells. So when is SETI?
Alessandro Sette?
Yeah, like next week.
I think the 20th or something like that. Don't
It's
remember.
like next week.
We have 3 immunology people this month. We have Sete, we have John Udell returning, and we have the head of the Vaccine Research Institute. What's his name?
Center.
What?
John Mascola.
Don Mascola. Should be interesting.
Yeah, I'd like to see what he has to say about precise.
About what? Precise? You want me to ask him about that?
Premise, not precise. Premise.
Premise.
Well, it's basically a recapitulation of my paper.
So the Pfizer vaccine is working quite well, especially if you give a 3rd dose. So the end of that. Rob, thank you for your support of the incubator.
Oh, that was the other thing that Offit said, that he doesn't believe in these boosts, that they're just going to give you an extra 3 months of high titer antibodies, and then you'll be back at the same level that you were before. And I agree with him.
Okay. Animal Party would like to hear us talk about our favorite scientist. My favorite scientist is Amy Rosenfeld.
No, it's not. Tell him who your favorite scientist is, really. It's like Peyton Rouse.
Really? I said Peyton Rouse was my favorite scientist? No,
Yeah,
I always liked David Baltimore, frankly. Who's your favorite scientist?
I don't know. I like a lot.
Just name one. Richard Axel?
No. I think Cahal is really interesting.
Who?
Cahal, the drawings of the brain and stuff is really interesting.
Oh.
He was really good.
The Spanish guy.
Yeah, the Spanish guy.
All right, let's get his full name. Cajal, right?
Yeah.
Santiago Ramon y Cajal, a Spanish neuroscientist, pathologist, and histologist.
Yeah.
And he died in 1934, was born in 1852. So Amy and I went to see his drawings of neurons and astrocytes and so forth at NYU Museum. Fabulous.
Yeah.
Fabulous.
And he didn't have a lot of technology, he just did it off of a regular microscope. No fancy EM, just a regular 4X or 10X light microscope.
Okay. Hospitalizations are up and many are vaxxed. Does vaccine need tweaking? No. It's up a little, yeah, but that's because we're having more infections now. The ratio is still the same. The protection is still in the 90s. When you start to get a lot of people infected, 8% makes a difference, okay? Don't make conclusions.
So Offit said that all the people who are in the hospital who need medical intervention or in ICU are the unvaccinated.
That's right. That's what Daniel says for the most part.
That's what he said.
Of course, it depends in what state also, but
He's
Texas...
in Philadelphia, but you know what? You should have the guy that he talked about today. You should have him on when you start This Week in Public Health with Laurie Garrett. So he went out to North Philadelphia where they don't really vaccinate. They're vaccine hesitant.
Yeah.
He set up a clinic there, and he got that whole area vaccinated. Like he talked to all of them about how to overcome vaccine hesitancy.
What's this guy's name?
I forgot. I'll look it up. But he's some colleague of Offit's, and he's some young guy. And I think it would be a great thing that we should learn. We should discuss how to overcome vaccine hesitancy.
Okay. Rasha wants to know, what would you do? Live in Florida, 8 and 5-year-old just got over mild COVID. 8-year-old vax, not 5. Husband and I are vax boosted. Cruise in March, 2 months post-COVID. Risky?
You live in Florida and you're vaccinated.
It's good.
Go on a cruise. Be my guest. Go on a cruise.
Is everyone going or just you? I guess you're taking the kids, right?
Well, they're vaccinated, right?
Well, the 5-year-old is not.
Why? The 5-year-old could be vaccinated.
The 5-year-old just got over mild COVID last week, so it can't be vaxxed for a while, right? All right, you heard it from Amy, and I'm telling you, Amy's right. Okay? All right, Zadius wants to know, recent paper about long COVID hypothesized autoantibodies against ACE2 may develop as anti-idiotypic antibodies to spike antibodies. If the infection can do this, can the vaccine? Do you know anything about this, Amy?
I don't know anything about it.
I think this is quite speculative. I understand there is some evidence for autoantibodies, yes, but I have not seen it with vaccines. I've only seen this, which talks about during infection. And I think its relevance remains to be determined still.
Yeah, I'm not clear.
All right. So they wanted to guess my glasses.
They're always blue.
Oh, someone asked later, it'll come up, when is your blog post appearing? So it was delayed a week because I had a guest post last week, but it will be up tomorrow. Right, Amy?
Okay.
Yeah, I'm going to put it up tomorrow. Virology.ws. Okay? Virology. Do you know how to spell that?
No.
.ws. Here. There it is. Virology.ws. Okie dokie. Can you explain, Amy, why we're sequencing too much? Oh, boy. Go ahead, Amy. Hello?
Well, what's the point of the sequencing?
Answer the question.
Can I explain why we're sequencing too much?
Yeah, that's what you say a lot.
Yeah. Well, I don't understand why we're sequencing. So it doesn't change whether or not you have Delta or Omicron. It doesn't change case management. You still go home and monitor your symptoms. And then if you have severe disease, you do something or other. You can easily make a TACMan assay to discriminate whether or not you have any of the variants in case you get monoclonals and you need a specific monoclonal. So you don't really need to sequence that whatsoever. So if you're sequencing to genotyped for treatment or whatever, it's not required. We're sequencing too much. Well, if you're sequencing as a scientist to understand how viruses change in relationship to adapting to a new host and under different selective pressures and under this condition and under this other condition and in different genetic backgrounds, that's a separate issue.
But what if you want to treat someone comes in with getting severe, they have comorbidities, they need monoclonals. Don't you want to know which ones to give them?
You can use a TACMan.
Okay.
Make a TACMan.
That's right. You don't have to do genome sequencing. So you can
just make a TACMan.
Okay. It's a PCR which can tell you if the changes are there or not.
Yeah,
okay.
It's what you use for HIV.
Exactly.
So what's the problem?
Exactly. Okay.
It's just laziness.
Amy, do we know how people with asthma are handling COVID? Is asthma a comorbidity?
Maybe for kids. I don't think it's clear.
For adults, you don't think it's clear?
Kids, you usually grow out of asthma at like around your late or mid-teens.
But still, some adults remain with asthma. I think it's a comorbidity. No,
it's really really obesity.
Pete wants to know if I'd do a SARS-CoV-2 lecture. Yeah, I could do a SARS-CoV-2 virology live, like a 4-hour lecture. How would that be? Sometime.
4 hours?
Yeah, and just take questions and talk about all the coronaviruses and what we understand about spillover and replication and et cetera, immunity. Yeah, I could do that. Sure. Not now. I have to start teaching at Columbia in a couple of weeks, but give me some time to get cranking there.
So like July.
Yeah, summer is good.
Except you're teaching what semester in the summer?
I haven't decided yet. It's going to be either the first or the second semester. I might want to do the second so that I don't teach immediately after the spring semester, you know? Mm-hmm. So Environmental Coffee House said so many people up in Buffalo getting COVID, it's overwhelming. People I know have died last week, hospitalizations, but my vaxxed friends are not dying. Yes, we have a lot of infections now because people are mingling a lot with the holidays and so forth. There's nothing special going on, okay? The press would like you to think that. Scientifically, virologically, there's nothing special going on. Okay, so you have said vaccine-infected people are mostly non-infectious and only 20% of infected transmit to others. Is still true for Omicron? Yes, it's still true for Omicron. How does this square with explosion of cases where percent vaxxed are greater than 90? In that paper I just showed you, the preprint, they found the viral loads are similar. The infectious viral loads are similar between Omicron and Delta. So we don't know why Omicron has a fitness advantage. And you have to be careful when you say explosion of cases in places where the percent vax is over 90%. I think those are very rare. And overall, it's much less than 90%. And then 10% of many millions of people is still a lot of people. And that's where the explosion comes from. And also, to make a point that Amy would, they're testing asymptomatic people. So the case numbers go up, and it looks horrible, but they're not contributing anything.
So what does this mean? I dismiss entire countries' approaches which have been determined by multiple experts.
Amy is an expert. Okay? And these experts are not virologists. You know, people. I talked to a journalist in Denmark, and he said, you know, we have an expert here who says you're wrong. I said, well, you know, maybe he or she is not an expert. I don't care.
Isn't Copenhagen in Denmark? Or is my
yes, it is.
Is my geography off?
No, it's in.
So isn't Marion there?
No, Marion is in the Netherlands.
Oh, Marion's in the Netherlands.
That's south.
Oh. Okay, well, my map is upside down.
Okay, with today's paper from Geneva, which we just mentioned, on low viable virus in those vaccinated, poor correlation, did Amy say, you think? Folks, I just want you to appreciate Amy and stop saying she's dismissing countries' approaches, because obviously she's bloody smart and gets it. And these dismissing is going to be the norm.
I mean, at the end of the day, they catch up like a year later and they come up with some ridiculous story to explain why they were wrong without ever having to say, I was wrong because I didn't really understand.
Yeah.
And then what happens is that then they lose faith and people don't want to trust them as their expert for the next question or the next wave or whatever. And then they boot the person out who hired them. It's a good plan. Works well.
All right, you often say they weren't designed to be sterilizing. Some may misunderstand that wording as an option deliberately not taken. People don't get that HPV was a one-off fluke. And that's the wording that Amy has in her blog post, something to that extent, right, Amy?
Yeah, yeah. I mean, for us to really understand how the HPV vaccine protects against transmission and infection is going to take years.
Barry, the reason I say this, and I understand the concern, because the bloody press thinks that contraction of antibodies is a bad thing and that we shouldn't be getting infected. They don't understand. There was a comment on one of the TWIV episodes today, and the lady said, TWIV has been hammering this for years, but the press still don't get that the vaccines don't prevent infection, they prevent disease. So I'm trying to emphasize that. So I'm sorry if the wording was not correct.
But it's not the press. The press talks to the scientists.
Yeah,
it's the scientists.
They talk to pundits.
Yeah, they talk to certain scientists over and over again, and the scientists weren't trained properly.
They should be talking to you.
I don't have time. I'm busy writing grants.
The world
trying to not get fired.
The world depends on you. How can you turn your back to them? I'm trying
not to get fired here, babe.
Yeah, I understand. All right, can you discuss the value of having one shot for a 5-year-old? Would you enroll your kid in school and in hockey with one shot given high rates of Omicron today?
Yeah.
Why is that? What's the value of one shot?
Well, if you wait long enough, it'll probably give you a great deal of breath. And eventually they'll give me a booster. I'm sure I'm going to hear in like 3 weeks that there's boosters for kids because there's already a booster for the 11 to 16-year-olds. So just give them time.
Is there a published list of all FDA-approved human vaccines demonstrated to be non-versus sterilizing? No. There's no such list.
You have to go and look through the literature and it's not in one paper. It's in like 60 papers.
Yeah, we're gonna work on this because this is something that I think the HPV is really the one-off.
For sure.
But I'd like to look at the other results. But see, the problem is most of the other vaccines were made at a time where we didn't have PCR to check shedding. So it's very hard to measure it. And people didn't do it. But we will try and take a look at that. Chris says he's grateful for Amy's uncanny ability to discern fact from supposition and hype. You bet. She can sort it out.
And actually, you didn't want PCR. So Offit says how actually they used to actually just give the swab and grow out virus so that they know how much is there. Because he's not a big fan of this PCR stuff. So yeah.
What type of lab tests are performed to determine the contagiousness and virulence of a virus? Well, you need an animal model, but as Amy is going to say in a minute, they're just surrogates for people. You have to understand that what you find may not apply to people. For example, we did a paper on TWIV last week with Amy on TWIV that showed that Omicron appears to be less virulent in mice and in hamsters. Who knows if that's the same in people? That we don't have the right data yet. So you do studies in animals to look at virulence, and you look at, if you can, at transmission or contagiousness. But not often you can't study transmission in animal models. The ones for SARS-CoV-2, only the ferrets and hamsters transmit. Is that right, Amy?
Yeah.
And so Tony comes up with the same question. Would you speculate it is for unvaccinated people? So ask Daniel. He said people are dying. People are getting severe COVID from Omicron. In his eyes, he's not seeing an attenuation in people. But I think it's still early, and we have to get a lot of crappy data before we get the truth. This is what Amy has taught me to do, to be negative, right?
No, I didn't teach you to be negative. I taught you to be
critical. Sorry. Yes, you're absolutely right. Recent studies touting CBD as blocking COVID.
Oh, really?
Yeah.
Oh, interesting.
I don't think that CBD. What do you mean? I don't know what it means to block it. It's not going to block. It's not an antiviral, if that's what you're saying. Oh, here's a good one for Amy. Do you think that R naught is a useful metric, and if so, how should it be determined?
I don't think it's useful at all. I don't believe in it. I don't think that there's any benefit to it whatsoever. I don't actually think it's a number. I think it's made up. You push some buttons on the calculator and it spews out some number, and then you give it a title.
So the problem with R naught is it's a mishmash. So the name is basic reproduction index. And there's no transmission in that word, if you notice. And it's a combination of 3 different variables, which include human behavior and the virus itself. And here's the thing, Jack. You can change, it's not R naught. When you determine it early in an outbreak, it's R naught. But then later, when you fitzle with it, as Amy would say, you can change it. You can reduce it, and if you can reduce it below 1, you can stop an outbreak. So it's a squishy variable, as Amy has said on multiple occasions. Right, Amy?
Yeah, but even still, even the virus part, it's not intrinsic to the virus because it can change. It can change. So how are you going to measure virus reproduction? Are you going to measure it in Aerosols, in Calo2 cells, in mice? How are you measuring it? So like it replicates at different efficiencies in different cells because of the host environment. So it's not an intrinsic property of the virus. So it's a useless number. It's made up to assuage us. And the truth of the matter is it's not an R naught beneath 1 that stops the outbreak. It's the number of cases per 100,000, and you want it to be under 1. 1 case per 100,000. One infection. Yeah.
So I can tell now that people are going to disagree with you, but just remember, Amy is usually right. Okay, so just hold back.
They can all disagree with me. That's fine. You can disagree with me if you. It's not. That's not the problem. The problem is when somebody writes, "She's just arrogant," because he felt that because he was wrong, he felt that it was not valid that he was wrong, and that becomes a personal attack. I'm not attacking him, calling him an idiot. Don't attack me and call me names. If you want to explain why you think that their opinion is more sound scientifically than mine, that's fine. I have no problem with that. But to sit there and say, oh, she's arrogant is ridiculous.
Don't pay attention to that. Okay, the breadth of B cells after infection plus 6 months of somatic hypermutation should be far greater than the breadth after vaccination plus 6 months, shouldn't it?
Well, are we talking breadth against every epitope in the virus? Yes. If we're talking about spike, probably not. It should be equal.
Perfect answer. Right on. It's perfect. Exactly right. What's the difference between immune evasiveness or contagiousness? Ultimately, don't both equal more spread?
No.
Immune evasion is one way a virus can be more fit than another.
But it doesn't equal, it's defined by whether or not you evade. What are you evading? Are you evading antibodies or
Mostly,
are you evading T cells? Most
Mostly
of
it's
it is
antibodies.
evaded, right. That doesn't mean you're more fit at all, because the T cells may be, the cytotoxic T cells may be more important in resolving viral infection than the B cells.
Well, it depends how long it takes, right? Because you may have a window where you can transmit, and the T cells haven't killed enough cells yet to tamp down.
Well, then your antibodies aren't there anyway.
Yeah.
Because your antibodies come after even the memory T cells. The CTLs come way before the B cells.
Anyway, I would say contagiousness is something different, right? But contagiousness could have multiple explanations. Immune evasiveness is one property, but contagiousness can have to do with that. It can have to do with the speed of reproduction, the titers that are shed, and so forth, stability in aerosols. What would happen if everyone wore N95s outside their house for 8 weeks? Would mailing masks to everyone be better spent dollars than testing?
Sure.
Thank you, Pamela, for your contribution to the incubator. This Ontario medical officer commonly says Omicron is more transmissible, less virulent than Delta. Well, he's just, or she is just repeating what they hear from WHO and other health agencies who, as you know, don't always get it right. So I don't think taking my course would help, although some of the principles would be very useful, I think.
What principle? What lecture is the principle of don't leave off the data, the description of the data set under? I must have missed that lecture. Is that in the methodology section?
What do you say? Are you saying I don't teach to depend on data? I think I do, don't I?
No, I'm saying that when I wrote the paper or I talked about the data, I left out the fact that in order to have concluded that I was less infectious or resulted in a milder infection, I forgot to include the fact that my population was mostly either vaccinated or had been previously infected by Delta. Okay. I just came out and said the isolate was less virulent or less pathogenic. So I want to know, in what lecture of your virology course does it tell me that I need to put out that my population really is people who were previously infected?
I don't explicitly say that. You're right. I'm teaching principles, Amy. And Mark says, I can hear Amy saying, come on, how can you make that call? That's right. And a cardigan has buttons. Thank you. Is that Mr. Roberts who wore the cardigans?
Mr. Rogers had zippers.
Mr. Rogers, sorry.
Fred Rogers, he had a zipper. Yeah, he wore cardigans and his sneakers. He changed it into his house shoes and he had zippers.
Well, if a sweater has a zipper, it's not a cardigan, is it?
It could be a cardigan. It's basically something that you have to put together.
You know, when it comes to this cultural stuff, I don't know anything. I guess this is not culture.
This is fashion.
This is fashion. I don't know anything.
Well, good thing your muse knows all about it.
All I know is that I love button-down shirts. I hate non-button-downs where the wings are flying. I just can't handle it. Those are supposed to be worn with ties, so I don't wear ties. I love the buttons here. All my shirts are button-downs.
Yes, I know. It's hard to find.
Um, what courses did you take as students which have aged the best and the worst? And I could say Amy took biochemistry courses that have aged the best, right?
I only took one biochemistry course as an undergrad. Um, I would say actually the neuroscience course, for me, the neuroscience course aged the best. It was basically fundamentals
Mm.
of like neurotransmission and the Nernst equation and, um, cell biology of how neurons work and how the brain is organized and stuff. The class that probably and developmental biology probably aged the best because
Didn't take any of those.
yeah, I took those
I took,
classes.
so I think organic for me really aged well because the principles of chemistry are the same. And I
Yeah, I
still use
hated
them.
organic.
You hated it?
Yeah, I had a very hard time seeing reactions, like,
Oh, I liked it
how
very
they,
much.
yeah, I was not good at it.
And then I took molecular biology with Stan Cohen. No, it's not Stan. It's a Cohen, but I don't remember his name. He was great. And a lot of that has aged well. The basic principles of molecular genetics.
Yeah.
Sylvia, thank you so much. Here's to you. I love you. Extreme and continued gratitude for all you do. Bravi. We'll have to listen to the recording. Yes, Amy is Johnny Mac. Thank you, Sylvia, for your contribution. Someone sent me chocolate, Amy. One of the course listeners, Jan
Oh,
from
that's great.
Arizona. She sent me 2 special bars of chocolate. I haven't
That's
tried them
great.
yet.
That's great.
Do you like
Chocolate is
chocolate?
good.
Do you want me to bring
Yeah,
you some? All right, I'll bring you
sure,
some.
sure.
Should we get boosted now or wait for the Omicron modified booster vaccine? Get boosted. No, don't
Well,
wait.
it depends on when. I don't really care about the Omicron. I really care about what the timing was in correlation to the last shot. I'm not
Yeah,
really.
it should be 6 months from your last shot. I think the Omicron specific vaccines are just a play by there's no scientific data that says we need them. Now, that could change, but I doubt it. Shouldn't the second dose of ad vector vaccines use a different ad serotype? I think Sputnik 5 does this, but not J&J or AstraZeneca. What do you think?
I'm sorry. What?
When you give ad vectored vaccines, should you
Yeah,
use a different serotype for the second shot?
that's generally the principle, but that's not what they're doing.
Yeah, apparently it didn't matter for the J&J and the AstraZeneca. To many people's surprise, they still got a response after the second and third doses. So it's not as important as we thought it would be. Yeah.
Mm-hmm.
McEnroe was childish. Amy is not. That's very nice. Was he childish, really?
He used to have temper tantrums on the tennis court. That's what he's famous for when he was a player. But he's grown up. It's now that he's married and he and his wife have like 7 kids between the two of them. He's matured. He's gotten it together. But he was a big baby on the tennis court when he played. But he'll admit it.
But Amy is not a big baby in the lab, I have to tell you. She's very professional.
No, I'm not a big baby.
Powell wants to know, can you comment on EU regulators saying frequent booster shots may be harmful? Can you comment on that, Amy?
How did they make that conclusion?
I don't know, but
Not
I'm
clear.
assuming they're thinking you're going to start making autoantibodies, as you get more and more somatic diversification.
Well, I'm not really clear that that's what happens.
I think,
I need more data before
yeah,
I make
I
a
think it's too
statement.
soon to say. There was some news that getting common cold offers better protection than vaccine. So I think that was the old discovery of Crotty and Sete. They said there's some cross-reactive T cells with common cold
Yeah,
coronas. But whether
sure.
they give you any protection or not against diseases is unknown. We have no idea.
Yeah, I mean, it doesn't seem that they do. I don't think that they would say that they did.
PCR is used to isolate a virus. How can it be validated as a tool? It's validating itself. Isn't that circular reasoning?
How is it used to isolate a virus?
It's not. You don't use PCR to isolate the virus. You put the nasopharyngeal swab in cell culture, and out comes the virus. Then you
Right.
sequence its genome, right?
Right.
And then from the genome sequence, you develop a PCR diagnostic test. So it's not
Right.
circular
And
at all.
and it's not circular, and it's not validating itself, because you do a series of titrations
Yeah.
and you do a series of negative controls, water and unrelated and tightly related viruses, blah blah blah.
Could it be possible if you don't mount a response to the vaccines, you would be more likely to get Omicron or any SARS-CoV-2? Yeah. Not everyone makes a response. Right, Amy?
Well, so the thing is that we're not really clear that they didn't make a response, since we only really basically test people for antibodies. They could have a very robust T cell response and be perfectly fine. All right, it's 8485, and I have to add overlay, so like 15 more minutes max.
So you're apparently Camille Pallia. Do you know Camille Pallia?
No?
She was a Yale professor.
Oh.
She studied sexuality, I think, but she was quite outspoken.
In terms of what?
That's the limit of my knowledge.
Oh,
You'll have to
okay.
ask. You have to ask someone else. It's a cultural thing. I'm not. I bought her book, but I never read it. It's upstairs.
Well, that's one step more than I did.
Thank you. They found Omicron in wastewater samples in Nova Scotia that predate South Africa detecting the variant. Is it possible Omicron originated in North America? What do you think?
It's just no. I don't think it originated in North America. I just think it was in the places earlier than was anticipated, and it wouldn't surprise me to find out that there are pieces of Omicron that were in the very first sequences where we found G614G, and then it just took it together, and then it took a little time for it to explode. Wouldn't surprise me whatsoever. I mean, that's the theory of Gary, right? Is that all of the variants were there at very low frequencies, and then they've exploded under certain selective pressures.
Can't wait to hear TWIV do the CBD prevent COVID paper. Okay, well, if it becomes a paper, we'll take a look at it. Thank you, Shane, for your contribution. Love what you're doing for access to education. Can you explain how COVID vaccines influence transmission and shedding?
They reduce it significantly.
Yes, you saw from the preprint we just mentioned briefly. They reduce shedding. And so that's what, and the paper did say shedding, viral loads, is one component of transmission. Not everything, which is another thing we've been saying for a long time, right, Amy?
Yes.
But the low shedding is not really consistent with high levels of transmission. So it's blunting transmission. The exact quantification remains to be determined. How protected?
You have to do contact studies after vaccination, and apparently we're never going to do them.
And you would have to do genome sequencing on isolates to prove that one person got it from the other instead of from going down to the drugstore, right?
Yeah, for sure.
And nobody's going to do that.
Oh, absolutely not.
How protected are 10-year-olds with 2 vaccines? The message in the media is all about how 2 vaccines aren't enough and boosters are needed. Depends how far apart they were. If they were 3 to 4 weeks apart, that's the problem. You don't give the immune response enough chance to mature, right, Amy?
Right.
So if they were 3 to 4, you should do a booster. They are not zero protected, though. They still have good protection against disease, but the booster would. Well, as you heard Paul Offit say, he doesn't believe in the boosters. But Amy and I got boosters. It doesn't hurt. I'm frustrated by media and admin use of breakthrough cases. It tends to diminish vaccines when everyone is getting breakthrough. Yes, we've always had a problem with the use of that term. It implies that something's wrong, and we discussed it on a recent TWIV. And then Kathy Spindler pulled out a 2018 paper which used breakthrough in the title, so it's nothing new to the pandemic. It's a bad word, I agree, especially in a pandemic. Thank you, John, for your contribution. Maybe it's a T-shirt. Never doubt Amy.
It's cute.
A 2020 study showed SARS-2 infected astrocytes lead to neuronal damage. I'm curious if more recent scholarship about the virus and its effects on neurons. This is Amy's area.
So what virus infects astrocytes? SARS-CoV-2?
Yeah, that's what he's saying.
No, does not infect astrocytes.
There's very little support for the idea that the virus reproduces in the brain or spinal cord.
There is no support for the idea that there is no support.
Okay.
We did several papers where they've looked at autopsies, and they have never been able to find viral RNA in any brain cell. Right. Or that's very, right. You know, I mean, it was like one cell over here and then like another 4 cells, 3/4 of a centimeter away. No. When you infect the brain, you can see it's clear. No.
So Amy,
it's unfortunate that it continues.
No, many people continue to push this narrative. It's wrong. Amy has a virus that infects astrocytes, and it probably causes neuronal damage. The activated astrocytes can cause neuronal damage. Right, Amy?
For sure.
Yeah.
For sure. I mean, that's one of the hypotheses for West Nile. Ken Tyler has shown that West Nile infects astrocytes and then can cause damage. Yeah, for sure.
What is the best interval between first and second doses of pediatric Pfizer?
Well, it would be like 6 to 9 months, but they don't really do that. I mean, I guess you could do that. You could forget to show up for your 3-week appointment and show up 9 months later.
Yeah, I would say 6 months, but you have to be careful, right? And minimize exposure in that time. Am I correct in that I'm on the wrong side of this thing?
I don't know. As I said, you're not. It's not a split screen for me, and I don't see the question. So there's something about whatever connection you have that is different today.
I can swap cameras, but now you're cut off. So actually, I like it better the way it was. We'll keep you on the left.
Okay, well, whatever.
Amy's blog post is going up tomorrow, as I said. Yep. Could we boost a 4th dose 3 months after the 3rd? No, you don't need to.
Welcome to Israel.
You don't need to do that. You don't need to do that. Are unvaccinated folks responsible for the variants? Who is? I think it's Amy, actually.
Yes, I spend my late nights when my cookies are baking or cooling. I come to the lab and I mix a concoction of goo and then I release it out the window. It doesn't open.
Our windows don't open. No, the unvaccinated are not the only. Why would you say that? No, I mean, they're responsible for lots of infections, but other people are infected. Wait a minute, Amy, what do you think? Let me ask you, are unvaccinated people providing the raw material for mutation and selection?
Well, the idea is that they're providing the raw material for the mutation because they replicate more virus, and therefore you have a better chance of having a more varied viral population, right?
Yes.
So
the answer is yes, in part, right?
Yeah, in part, but there is still, a selective force, and there's also things that occur stochastically.
So if most people were vaccinated and there were very little infections, there'd be fewer variants generated, correct?
Yes.
Yeah. And we wouldn't be having, most of you would not be here if 90% of the world were vaccinated right now. But the others, the 10% of you would be here, and we'd have a great conversation anyway.
Yeah, but about different viruses.
Roger said, if the mRNA vaccines are based only on spike, why don't T cells lose effectiveness like antibodies do?
Because their epitopes don't change.
The T cell epitopes do not change. And the reason for that has to do with the polymorphism of T cell receptors and major histocompatibility molecules. What do we know about the mechanisms of viruses used to cause neurological damage? Well, what's the lady neurologist at Columbia that we had on TWIV?
Karen Thacker.
Kieran Thacker said it's a combination of hypoxia and cytokines, not virus reproduction.
And Carlos has also said it's not virus reproduction too.
Are mitochondria involved with viruses? Can having a mitochondrial defect make you more susceptible?
Yes.
Well, really?
I think so. I think there's a few. I think that there's a little data that says it. So, like, the mitochondria is critical for the antiviral response. Like, what is it, MAVs that's docked to the mitochondria and stuff?
Oh, that's right, yeah.
But it also is a balance because the mitochondria is energy and viruses need the energy from the cells and other byproducts that occur in the mitochondria for nucleotide synthesis and stuff. So.
And of course, viruses require energy that's produced by the mitochondria, right?
Right. So.
I quote Amy and Vincent on Twitter and gleefully accept the wingnuts' crazy reactions. Where did this phrase wingnuts come from?
I don't know.
I know what a wingnut is, that little thing you can flick with your hands, but I don't know why.
Yeah, I know what a wingnut is.
You do?
I do.
Where can you listen to Paul Offit? Which podcast was he on?
He was on Slavit today.
Andy Slavit.
He was on In the Bubble, but he's been on Twiv like twice already. The last one was right before Christmas.
Yeah, so yeah,
he basically said the same thing, but he does it in more detail on Twiv. So if I wanted to listen to him, I'd have listened to him on Twiv.
Yeah, listen to him on Twiv. We go into it in more depth for sure. More and more studies are showing that T cells are kicking the viruses. Was there something that made you guys think neutralizing antibodies may not have been key? Did you know viruses had tucuses, Amy?
Oh, I did not know. Are they big and round and plumpy, or are they flat and squishy?
I don't know.
And do they come as saddlebaggy or no saddlebags?
So,
because I want mine to not have any saddlebags.
I understand. Here's my feeling, Mark, that when the first variant arose, Sete and Karate published a paper saying that T-cell epitopes had not changed. And that made it quite clear that the vaccines were still going to work because of the T-cells. And with every variant, they've said the same thing. The epitopes are barely changed. The T-cells still work. And so I am thinking more and more that there's a very reduced role of neutralizing antibodies in these infections. It's
well, historically, T cells aren't what clears coronas. B cells and antibodies. Historically, it's the importance of the T cell.
Yeah, I mean, certainly if
we
you have
wrote a whole grant on that.
Certainly, if you have a high antibody titer, it will block infection. The antibodies.
Yeah, but that's for any pathogen.
Yes, it's an artificial situation. Yeah. I saw a quote from Offit in The Atlantic. He wouldn't recommend a third dose for a 17-year-old, not for his son in his 20s. I didn't know that. Why is that, Amy? Do you know?
Probably so males at 17, and since they're only for Pfizer, that happens to be the age group that has the risk of myocarditis, and he doesn't. And he says that you really don't need a booster because all it does is give you 3 months of protection against mild disease, and that's not what the vaccines are supposed to do. Vaccines are supposed to protect against severe disease. All right, one more, and then I have to go because it's 9:00 and I need to add overlay.
Do you think that people realize U.S. taxpayers are funding about $100 billion annually for COVID tests? Is this a wise use of limited capital?
I don't think people are focused on how the government pays for the money because I think they think that the government is broke. Until we have overcome vaccine hesitancy, yeah, I think it's a fine use to send people tests or who are unvaccinated and have them sign up to get tests is fine. Testing that vaccinated is not necessary, but you can go, I don't know, And if you're unvaccinated, with your little credit card to the pharmacy and buy a bunch of tests, I think that's fine.
Okay. Thank you, Amy.
My pleasure. All right.
I'll
I
talk
don't
to you
know what's
tomorrow.
on this. Yeah, I forgot what's on the schedule.
I'll see you
Oh,
Friday.
you have a lot, like 15 podcasts.
Tomorrow, yeah. Well, not 15. I have Daniel at 3, Twip at 5, and then I have to set Daniel up for his own thing at 6. So just 2 pods. But we're going to have 4 people in the incubator for Twip.
Okay. Well, as I said, I guess we'll just discuss everything on Friday.
Thank you, Amy.
My pleasure. Have a good evening.
You too.
Bye.
Bye. Do I have any investments in biotech? I don't know, because I don't handle. I mean, my IRAs. No, not IRA. My retirement funds are invested, and they're funds, right? So there might be some biotech mixed in there, but I certainly don't look. And I don't call the guy and say, buy this guy. I don't do that. I don't have time for that. I don't really care much about money. Okay, I just like to teach people virology. I like to talk about viruses. I like to write about them. I like to make videos. I like to teach. And this is a problem because it's not responsible to not care about money. But anyway, I would not say buy this or that biotech or this or that pharma. And I'd rather not, because I always talk about these companies. I think it might be a conflict of interest, right? The data showing negative efficacy for the vaccine. Is it fake news? Yeah, of course it's fake news. The vaccine works very well against Omicron. Absolutely. If you don't test asymptomatic people, then we never detect them. So XXX Passy, you don't think Amy's right. Okay, fine. Well, she's gone. We never detect them. People keep saying we can't get reinfected. All right, look. Now the next set of data is we have to see how well vaccinated infected people transmit compared to unvaccinated infected people. We just don't know that. It's all been done by PCR and some epidemiology, okay? So we don't know. And I suspect, given the viral loads that we saw today in this paper that we discussed at the top, they're not likely to transmit, if anything, maybe in the first day or two, and then it rapidly declines. So it's going to be a big hit compared to unvaccinated people. So I think it's not a case for testing asymptomatic people at all. It doesn't do anyone any good. It doesn't change your treatment. I understand you're talking about transmission, but I think the role is minimal there. Do we have any studies showing the typical size of the particles? Yeah, I mean, there are electron micrographs where you could, they're pleomorphic. There's a range of sizes, and the numbers are something like 80 to 120 microns in diameter. So in my view, the issue is that the aerosols, right, the very fine droplets that can travel long distances, they're small. And so they can contain fewer virus particles of any virus. And so the key question here is, how many particles do you need to start an infection in a person? And we don't know. And if anyone says they do know, they're wrong, okay? Because there's no data on that. There are no good data on that. And it's a very hard experiment to do. So I suspect that in those aerosols, there's not enough virus to infect people unless you're a super producer. And as we've said many times, 80% of transmissions are caused by 20% of people who are infected. They make the most virus. They have at least the highest RNA viral loads. Nobody's measured their infectivity. So that's a good point that needs to be done. So I think these are good questions that we don't have answers to. In the context of a pandemic, it's very hard for this basic science to get done. I mean, this paper we discussed briefly is just fabulous. They did all the right things. And as I said, it took this long to do that. It's crazy. I'm currently 22 weeks pregnant, had COVID in December, got vaccinated in April, both doses, 4 weeks apart. So you're infected and vaccinated. I don't think you need to get boosted. You could ask your doctor, but that's what Daniel would say. If you were infected and vaccinated, your immunity is very good. And so don't pick on the moderators either. They're trying to do the best where we have a lot of people that are... I don't want people making comments about Amy saying she's this or that. There's no place for that in here. So if you did, you got deleted, I'm sorry. Maybe you're timed out. So just give them a benefit of the doubt. Amy being consistently accurate will never win her a spot on CNN. Yeah, I know. I was on CNN once in February 2020, and they never asked me back. I guess they didn't like the truth. They want more hysteria. Did Amy catch Andy Slavitt and Laurie Garrett on the Al Franken podcast? Interesting discussion showing how even a smart person like Al can get confused by the mainstream media narrative surrounding. No, she didn't mention that. Didn't mention that at all. Can my 3-month-old baby get COVID? Yes. Daniel has mentioned babies who are, it's relatively rare, but yes, you should be careful. If the sister is positive, she should stay away from the baby. What's the name of the paper that I put it up at the beginning of the stream? Let me put it up again. There it is. It's not a paper, it's a preprint. I think it's in one of the most important papers we have seen so far. Infectious viral load in unvaccinated and vaccinated patients infected with SARS-CoV-2, wild-type Delta, and Omicron. It's on medRxiv. So you can just Google that or go to medRxiv and infectious viral load. You're not going to find infectious viral load on other papers because they just don't do it. Yes, the paper's dynamite. I'm amazed they didn't do this for the 500 papers that were written without taking infectivity into account. I've been saying for years, and so has Amy, PCR is not infectious virus, but yet people still do it. Why? Because it's easy. And it's okay if you're doing diagnostics. No problem. But if you want to do an experiment, if you want a biological answer, you have to look for infectious virus. Bottom line is, a virus transmits when it's infectious, and if you're not measuring that, you're not measuring transmission. How about testing before you go see your 94-year-old mother-in-law? So Amy and I differ a bit. So, yes, I think if you have a 94-year-old mother-in-law, you don't know, even if they're triple vaccinated, you don't know how well they responded. You might want to be tested. The problem is, what does the result mean? So you could do a PCR test. It's probably too sensitive. It's going to pick up bits of RNA long after you've stopped transmitting. You want to do an antigen test. So the assumption is the antigen positivity means you're making virus and maybe enough to transmit, but you don't know. So I think the default would be if you did an antigen test and you're positive, don't go see your mother-in-law. I think that's a reasonable use. But I don't think it's reasonable when people are just, hey, I'm going to see if I'm positive. Or they're testing students on campuses who are mostly asymptomatic. I don't think that's useful. Amy, I didn't understand your comments on Twiv about small children not responding to the Pfizer vaccine. So she just mentioned it again tonight. So it's a preprint, I believe. So you're going to have to go check that out because I haven't looked at it. Bottom line, allow competent scientists to do their job. The problem is mainstream media wants to report it, right? That's how they make a living. So most of the time they're doing politics and economics and other things, and very rarely infectious diseases, right? But then when there's a big one like this one, they get in the act because they want to make their money, and they screw it up. There's no question. And I'm not saying everybody, they're good ones and not so good ones, but there's subtleties, there are nuances that are not appreciated. And I do wish that we had more of a reach, but a lot of people tell me you have a good reach because we talk to people about what we hear on your shows and it gets transmitted. So that's great. Secondary attack rate is, so you have, typically you can do this in a household, right? You could have a person who's infected and then see how many people that person infects, right? And it's a measure of transmission, because spread of, it's basically the transmission in a circumscribed group. It could be your house, it could be a dormitory, it could be, even a lecture hall. And they do that as opposed to saying, looking at transmission in the general population where you don't know who's infected whom, right? So that's a good way to get an idea of how well the virus is spreading. It doesn't tell you the intrinsic transmissibility of the virus, though. But people use the word transmission in different ways. I use it very differently. They're using it in just spreading. Transmission, to me, is a very specific virus property. What they're talking about is fitness of the virus, and that's driven by a lot of things, including the virus, how well it reproduces, and other things, immune evasion, and the population, right? Population getting together. Amy was right. Well, we do have a Right Amy T-shirt. I think we should have an Amy was right T-shirt. That's true. Results from protein-derived vaccines anywhere. So, the Novavax, we're still waiting. I haven't heard anything. Of course, the Baylor vaccine being produced in India, the protein-based vaccine, I'm very worried about that. They're not measuring the ability of the vaccine to prevent COVID. They're measuring the ability of the vaccine to induce a certain level of neutralizing antibodies. And I don't think that's good enough, because I'm thinking antibodies have a minimal role, especially when they begin to contract, right? So maybe it doesn't do a good job with T cells. Anyway, so that's in the trial. We don't know the results yet. It is absolutely useful to have a discourse. However, when it gets personal, it's not useful. And that's what she's talking about, all right? And Amy and I have discourses all the time, but we don't insult each other. And unfortunately here, people insult her, and it doesn't work out. And, it's not useful for you to say you're wrong, especially if you have no basis for saying that. It's just your feeling, or you heard it in the news or something like that. All right, Mark, you're on fire tonight. Last time you were pretty quiet. From an objective standpoint, is the virus interesting? Is there anything very different or cool about it? Yes, it's quite interesting. Many aspects of it are quite interesting. The fact that it was able to spread so widely, even at the start. Remember, before any variants arose, this virus spread really well. It just got better because of fitness improvement. So that's very interesting. We've never seen that before in real time. I think the way it leads to severe COVID and long COVID is really interesting. So they're very interesting things, but most of them have not been explored. Most of the work done is just rediscovering fundamental principles of immunology that people seem to have not known to begin with. There was something. Oh, here you go. This one. What's the exciting lab result? So Amy and I have a paper coming out next week in MBio showing an antigenic relationship among enteroviruses. Enterovirus like 68, 71, 70, rhinoviruses, polioviruses. So we'll have her on TWIV when it comes out to talk about it, because I think it's really exciting, and it's not something anyone ever did or found before, and I think it's important. I'm 2x AstraZeneca, 1 Pfizer booster. I have kidney issues, arthritis, and obesity. Am I safe to catch Omicron, or do I shield? Well, look, the protection against severe disease is in the 90th percentile. But here's the thing, and Daniel Griffin tells me this. The people who end up getting sick, the vaccinated people, even the triple vaccinated people, and others have said this as well, the people who end up getting severely ill after triple vaccination are typically ones with comorbidities, right? And so obesity is a comorbidity. So I would say you should be careful. Amy still believes 5 years total. Yes. If virulence is largely dependent on a host response, is it to say X is intrinsically more virulent than Y? A meaningless statement. So virulence is very difficult. We have a whole lecture on this in my virology course, all right? And it's really worth listening to. It's called Infection Basics. And essentially in the laboratory, you can measure virulence. So there's 2 ways you can measure it in people. I mean, you can't really measure it because you cannot do an experiment in people. But in a lab, you can take an animal that gets disease after infection. You can give it a certain amount of virus by a certain route of inoculation, and you can measure signs. Right? Symptoms are not, we don't do symptoms in animals because they can't tell you my leg hurts or my head hurts, right? You can measure things, and those are signs. And then you can compare the virulence of 2 polioviruses, for example. But you can't compare influenza and Ebola virus because they're 2 viruses and they do different things in the animal. And you would need different animal models on top of it. So it's not relevant to say, even though you feel that, Ebola is more virulent, right? It seems to kill more people, but that may not be all intrinsic to the virus, because Ebola virus infection tends to be more lethal in Africa when people don't get good health care. When they get to other countries where they can get the proper care, they survive. So that's the problem. And you can't do the experiment in people. Anyway, it's not only dependent on the host immune response. Of course, if you let a virus reproduce unchecked in an animal, it will kill it. Most of the time, if you take away the interferon system, most viruses will kill the animal. So there's a big role for the immune system. And in fact, many signs of infection are caused by immune responses, but there's certainly a part of the virus because you can make mutations in viral genes and decrease the virulence of a virus. And not all of those genes are immune antagonists. Those are some of them, but there are other genes as well that control different aspects of viral reproduction. Anyway, this is a really interesting topic, and I really recommend Infection Basics. Is that the one? No, that's not the one. Mechanisms of Pathogenesis. That's the lecture where we talk about this, okay? It would be worth your while. I think you'll find it illuminating. I love teaching about viruses, and it makes it interesting, I think. Oh, what's the title of the paper you mentioned? And questions are tricky. What's his name? Nevin. I know. Kroger, I think is his last name. I should know, but I forget names very easily. Let me see if I can look it up. Nevin Kroger at UCSF. I know he's at UCSF. Krogan. Nevin, Nevan. Krogan. Krogan. He is at the University of California, San Francisco. And he published a paper showing what Amy said was that the other changes in the variants are more responsible for their different properties than the spike changes. Yep. Questions are tricky. Is it likely that infection with Omicron after vaccination will produce better cross-reactivity against future variants than infection with any of the variants less mutated? I don't think so. I think any of those variants makes a good broad response. But of course, that's a supposition on my part, and we'd need to look at it, right? But I don't think so. I think they're all good at making a broad range of antibodies. What kind of risks are we looking at from the massive animal reservoirs that are allowing the virus to continue mutating? Well, not just mutating, but spreading, right? The main risk is that you'll never get rid of SARS-CoV-2. It is here forever. If it were exclusively a human virus like SARS-CoV, we could eliminate it, which we did with SARS-CoV. Now, you could argue that an ancestor is in bats, but that is most likely a different virus. So that's the problem. We can't get rid of it, so we're stuck with it. It's another human virus to add to the list. Now, the other issue is, of course, it could change in the animals and get back into people and be worse off. And maybe it could be more immune evasive or something else. I doubt it, but that's why we have to do wildlife surveillance big time. We're not doing enough of it right now. We need to do more. Dr. Ginberg said Sputnik 5 doesn't require adjustments for Omicron. So that is correct. None of the vaccines need adjustments because it's the T-cell epitopes that protect you in the end from severe disease. Remember, the vaccines are not preventing infection. They're allowing infection, but they're preventing severe disease. This is really important, and we can't repeat it enough. So with every variant, the antibody epitopes change, and so the neutralization drops. But the T-cell epitopes are not changed. The T-cells can still kill infected cells, and that keeps you from getting sick. And that's why Ginberg is right. It's not propaganda. He's absolutely correct. The Pfizer CEO saying you need a new vaccine is not right. Right now, today, we do not need new vaccines. Can you elaborate on ORF9B relationship? No, I don't know enough about what it's going to be doing, right? And Amy might be able to do it, but I can't. It's an immune evasion protein, right? That targets mitochondria. But I don't know what the implications for pathogenesis are. If there are changes in it, it could be less immune antagonistic, right? It could attenuate the virulence. And maybe that's what we're seeing in the hamsters and the mice with Omicron. But I don't know about people. You can't extend it automatically. It's a really dangerous thing to do, to see a result of an animal experiment and say this is what's happening in people. Can't do it. Do you think the virus passes through discrete? Well, it should be discrete. I don't know, discrete or discrete, the two different spellings, two different meanings. I mean, discrete could be surreptitious, right? Or it could mean contained places and then leaves, like an apartment building where the floor where I work had a flurry of cases and then gone. I don't know if you're meaning the person or in a building. Yes, in a building for sure. There are a lot of factors that control who would get infected. That's why if someone walks in a building with 100 floors, not everybody gets infected because there are a lot of variables, right? The virus is not going to, unless the ventilation is circulating virus throughout the building, is not likely to survive infectivity. And maybe the people are, not all people transmit. They may not all go from floor to floor. So yeah, it tends to be compartmentalized. And then the virus infects a number of people, 50 people, and then it ends. It can't find a new host. You know, it has to be in the right place at the right time. It's a lot of chance involved. It's not a given that it's going to find every susceptible host. It doesn't work that way. If you could remove one COVID-19 restriction that is enforced now, what would it be? I think, so I dislike the quarantine isolation, especially for asymptomatic people who test positive. I just don't think they're transmitting. There's no data that they are. I understand that it's in the interest of safety, but my feeling is we have multiple vaccines that work. They prevent you from getting really sick, and it's time to return to life. If we have a lot of people that don't want to be vaccinated, they've made their choice. Now, the problem is that they get sick and it burdens hospitals, so it's not fair to the hospitals and the healthcare system. But I'm in favor of getting back to, I don't think closing universities makes any sense. Columbia's going 2 weeks online next semester. It's ridiculous. There's just no reason for that. So it doesn't make any sense. So Amy said earlier, the longer the better, right? Because currently it's 3 or 4 weeks. But in adults anyway, 5 to 6 months is better for the third dose. And so you would think between the first and second, 5 to 6 months would be better. But you have to balance that between the risk of being infected if you just have the first dose, right? So I would say if it were my kid, I'd give him the 2 doses 4 weeks apart and then a third dose 6 weeks later, 6 months later, just to avoid that period when you're particularly susceptible. A patient's long COVID lasted more than a year, got better after Omicron. Well, this was also the reporting when the vaccines were starting to be used. That I think about 30% of people with long COVID said certain of their symptoms regressed, right? So I don't know what to make of that. It needs to be studied, obviously, but again, in a pandemic, it's hard to do the kind of studies that are necessary. But I think, yes, it would give you insight. I'm not sure exactly what it's telling us. I mean, if there's some dysregulation of the immune response in long COVID, then maybe vaccination or infected sets it in the right way, but it doesn't work with everyone. It's only a subset of people, so not clear what's going on. I read an article that said Moderna offers more protection than Pfizer. So Daniel mentioned that last week as well. He suggested that there was a slight advantage, but he thinks it's not. I mean, he didn't think it was worth getting one over the other as a consequence. So he thinks it's very small. So I don't think it's hogwash, but I think it's really minimal. And I don't know what the reason for that, if it's the dose or something else, right? It's hard to know without doing the right experiments. How will we know when the virus is endemic? Well, endemicity means the virus is always in a population, right? It's endemic. It's here to stay. I mean, people are talking about, when is the virus? It's endemic. Influenza is endemic, right? It comes back every year. We have COVID every year. We're going to have it every year. And it's started. So I would say it is endemic because it's not going anywhere. What's a bigger concern going forward? Novel corona pandemic unrelated to COVID-2 or emergence of a variant that substantially evades current immunity? We will not have a variant that evades current immunity. No. It's not happening. I think the T cells are handling this really well. We've had a huge number of variants emerge, and none of them have gone around the T cells. I think a new corona, COVID-3, bats have a lot of viruses, especially coronas. So we have to, we don't study them enough. We don't really know what's out there. So I think that's the bigger threat. And don't forget influenza. That's going to be the next pandemic, most likely. Australia has reduced the interval between second and third doses to 3 months. I think that's wrong. I think 5 to 6 months would be better based on the Ontario study. I don't see where the data comes from for them to do that. I think we talked about this last week. Not a good idea. Is the loss of taste, smell from COVID a neurological symptom? Yes, it is. As far as the smell goes, you have an olfactory epithelium up here, right? And the chemicals bind receptors in your nasal tract, and those receptors are wired to your brain, to the olfactory bulb. And there are cells around the neuron, the neuron termini in your olfactory epithelium. called sustentacular cells. They're support cells, and they're infected by SARS-CoV-2. They're damaged, and that's why you can't smell and you can't taste. Okay, that's the mechanism. That's been really worked out very nicely by a number of labs. And we had early on a guest on TWEVO who talked about that, sustentacular targeting by the virus. And recently we did a paper on TWIV where they took patients who died right after them dying and pulled out pieces of the olfactory epithelium and saw the viruses in sustentacular cells, not in the neural cells. If you need 3 doses for the vaccine to be effective in protection from being infected with Omicron, how are 5 to 11 kids covered if they only have 2? Well, so here's the problem. The 3 doses are needed because doses 1 and 2 were spaced too closely. In the Ontario study that I keep citing, showed that this 3rd dose corrects for that. So if you did doses 1 and 2 at 5-6 months, you would be okay, but they don't want to leave the kids unprotected for 5-6 months. So eventually they're going to probably get a 3rd dose unless we don't see increase in disease in that age group. That's going to be from observation. Lynn, thank you for your support. My 20-year-old healthy, no-risk son had COVID before vaccines. He had 1 dose. No, you're all set. Recovery and 1 dose, you're all set. A 2nd dose doesn't help in broadening your immune response. Oh, by the way, we welcome our new moderator tonight, Tom, who reminds you, if you want us to answer a question, preface it with A or V. That's because you guys are having good conversations, and I don't need to interrupt them. And here's another CBD question. So I need to look at these data. I suspect they're anecdotal and not based on science, and that's what we're all about here. Still confused about the 3rd shot, if it just raises neutralizing antibodies or does more. So it does 2 things. It does raise the neutralizing antibodies, but that's temporary, right? That's going to go back. They're going to go back down in 6 to 8 months. But what they do that's different is they broaden the antibody response. So if you just had 2 doses at 3 to 4 weeks apart, your serum has trouble blocking infection with Omicron. But if you get a 3rd dose now 6 months later, your serum can handle Omicron. So in my view, that's the best rationale for the 3rd dose. Now, it still might be that even though your 2-dose serum can't handle Omicron, that you're still not going to get serious disease because your T cells are still there and their recognition sites are not changed. And the answer to that question we don't have yet. But we'll soon be able to say, okay, people who did only get 2 doses, how are they doing with Omicron? Those data will be out. So that's the justification. And when I saw that paper out of Ontario, I said, okay, this is worthwhile to get a dose because the first 2 were too closely spaced in terms of antibodies. Antibodies only. And I tell you, we don't know anything about T cells. Hal wouldn't have been able to do his magic if it weren't for the Golgi stains. That's right. Everybody, I stand on the shoulders of giants, right? Everyone stands on the shoulder. Whatever someone does that's good, it's always based on what someone else has done. And to not admit that is disingenuous. I'm supposed to work on Monday driving a truck with a coworker who's a positive symptomatic son at home. Am I at risk if I spend hours? So you don't know if he is infected, right? Have him take a test. And, even if he's infected, he may not be transmitting. So there are lots of circumstances you could both wear masks, and that would really cut down the risk. And yes, there are a lot of great scientists out there. Unsung scientists quietly working at what they love. That's what I did for years before I started podcasting and blogging and videoing. Yeah. It was very satisfying, and no one knew about it, but that was fine because I got to make the discoveries first. I knew something before anyone else did. Local health officer seems to be listening. Her public announcements, like still recommending 6 months to has pleased. Oh, good. I'm glad. I mean, that's not going to be everyone, right? Because everyone has different sources. But I think that's the key here, 6 months. Mike Rowe. I don't know Mike Rowe podcast. So today I was supposed to be on the McEnroe podcast. It's not John McEnroe. It's another McEnroe. Colin McEnroe from Connecticut. I've been on that before. I guess it's a radio show, right? It's not a podcast. But I didn't see the email till this morning. They wanted me at 1 p.m. You know, the night before they say, can you come on tomorrow? Why don't you give me a day's notice? I don't check my email like every hour. I'm not crazy like many people are about that. What's an overlooked or most understudied aspect of this virus? I think the reason for the increased fitness of variants, why they are able to displace other variants. You know, I mean, Delta looked pretty good, right? It seems to, and people, it seemed, according to this study, infectious virus shedding of Delta is greater than the ancestral virus. And so that alone could contribute to its increased fitness. But of course, in cells and culture, the virus doesn't grow better than ancestral virus. So I don't know what's going on there. So I think that's an important question that's going to take a while to sort out. Why do the variants have a selective advantage and displace others? We don't know. And if someone tells you it's about transmission, that's just not right. We don't know the mechanism of it. Last week you mentioned everyone you know has COVID. They're all fine. Nobody I know has been severely ill because everyone I know is vaccinated. The circle of people that I immediately, and I don't interact with many people. I do here and, you know, on the internet, but I don't do a lot. So I have my immediate family and then Amy, and some colleagues at work, but the incubator's empty. I go to the bank now and then, to the supermarket, but the people I know are all well. They're absolutely fine. My opinion on why hospitalizations are up, because the number of infections have gone up, right? So even if hospitalizations are 5% of infections, you know, if you have more infections, they go up. Because why are infections going up? I think it's the holidays, and people are now getting together more than they have in a year and a half, right? People feel, I'm vaccinated, it's over, I'm going to do my thing. In New York, you can go to restaurants, you can go to the theater, you can do almost anything. You just need to be vaccinated. So people are doing that, and there are a lot of unvaccinated people sneaking in there. They're getting infected, and that's why the hospitalizations are going up. There's a slight rise. But as I said earlier, I think that's just a numbers game. The percentage is the same. When a vaccine is 90% effective, what does that mean for the other 10%? So it could be that they didn't respond or they responded poorly. You know, there's not a black and white response to a vaccine, all or none. There's a whole gradient from really good to intermediate to low. And so in any population, there are non-responders and there are poor responders. And those are the ones that are going to get infected, even though they're vaccinated. And as I said earlier, it is most likely that those individuals have comorbidities, which, you know, if you have a slightly suboptimal immune response to the vaccine and you have a comorbidity on top of it, that's bad. Because if you were healthy and you got infected, you would be fine. But because they have a comorbidity which increases their susceptibility to severe disease, then it's a problem. So that's why not. And you know, 90% is bloody amazing. You got to understand that. You don't have any other vaccine that is so good at protecting people from severe disease. The polio vaccines, when they were first released, were 55% effective at preventing polio. So it's really remarkable, and no one could have predicted it, right? Nobody could have predicted it. It's not just the mRNA vaccines. The adenovirus vaccines are good. The protein-based vaccines are good as well. Yep. This is a good question. This is a perfectly reasonable question. Does the contraction of antibodies portend a drop in vaccine efficacy against severe disease later in time? I don't think so. I see no evidence for that, because we have seen a drop, a series of drops with the neutralization of the variants, and we have not seen a constant increase in severe disease. Matter of fact, it stayed, as Paul often said, it stayed at 90-some% protection across all the variants. Now, the powers that be in the U.S., and I don't really know who makes the decisions, decided that it was going to change, and that's why they advocated the third shot. But I think the real reason is to fix the defective timing of the first and second doses. That's a far better way to sell a third dose than to say, we're going to have an increase in severe disease. They had no reason to do that. And let me tell you what one of the reasons was they said that. Do you remember the CDC study out of Provincetown, Massachusetts, where a lot of people got together and some of them were vaccinated, some of them were not, and there were a lot of infections. They did some PCR, and they found in Delta, these were Delta infections, that the vaccinated people had as much PCR RNA as the unvaccinated. They said, oh, this is bad. They're getting infected. They're going to transmit. So we have to boost to prevent this. Well, it turns out that, as this preprint that I just read to you at the top said, you can't do a single time point, and you can't do PCR if you want to measure shedding. PCR for shedding is irrelevant. It doesn't correlate with shedding. So the CDC was completely misinterpreting those data, and they should not have made, and we said so at the time, this could be wrong. And there was a parallel study out of Singapore where they did time points, and they showed, even though it was viral load, RNA viral load measured by PCR, very rapidly after infection, the viral loads decreased in vaccinated people compared to unvaccinated people. So that said, something's different. There's some nuance there that they didn't pick up in the Provincetown study. So I don't think it portends a rise in severe disease. The only way to battle vaccine hesitancy is to listen to them. Yeah, I agree. I don't want to ban people. I try and talk to many people as I can, and don't get, the problem is they tend to get mad, and I don't want to have mad. Oh, tell me, what's your hesitancy? What is it? Oh, well, it's an experimental vaccine. Well, here's the reason why it's not. Well, it's a new platform. So we can talk about all that, and then in the end, if you don't believe it, that's fine. But if you're going to base it on political motivations or religious motivations, that's not good. It has to be based on science. So if you can tell me the scientific reason, and then if you decide in the end you don't want to be vaccinated, that is your choice. But I'm not banning you for sure, but obviously some institutions will ban you. How is Despommier doing with COVID? Oh, he's fine. He had a very mild little bit of hoarseness, no problem whatsoever. Despommier is fine. Other people at Columbia test positive. They're all fine. They're all vaccinated. They're all fine. Quick summary of the different types of vaccines. You can go to vaccines lecture. Well, basically, we have mRNA vaccines. We have 2 of those. We have adenovirus vectored vaccines. We have quite a few of them. The J&J Janssen here in the U.S., the AstraZeneca in Europe mainly, in other countries, and Sputnik in Russia, and there are others, other adenovirus vector. There's some inactivated virus vaccines where you take the virus, grow it up, and inactivate it with formalin. There are protein-based vaccines. And then there are some others that are experimental still. They have not yet gone into clinical trials, like attenuated vaccines. Those are still in the development phase, and I'm not sure how far they're going to get because we have ones that really work well, right? Yes, while you're at it, you've got 1,000 of you here, which is great. Third week in a row, we have 1,000 or more people. Give us a like. 461 likes. Why do I want you to like? It's not for my ego. It is to spread the word. And that is the honest truth. I want more people to find out. And if you give it a like, it's more likely that more people will find it. And here you can get good information. Would the incubator be able to conduct virus elimination testing? No, the incubator is not going to be a lab. The incubator is a recording studio, an office, so we're not going to do that. I love working at the incubator. It's my second career. I rarely go to Columbia now. Twice a week I go. Spend most of my time at the incubator. And if you want to come by and say hi, you're welcome. The success of the polio vaccine is the basis for my hope in vaccinations. I think my mom raised money for the March of Dimes when it was still working. Oh, yes, great story. March of Dimes fundraised. The 2 polio vaccines were funded entirely by private and philanthropic donations. No NIH support at all for the polio vaccines. It's great. Let your people know about your patron. So recently, Microbe TV became a 501c3, a nonprofit. So your donations are tax deductible. And that goes back to April of 2020, if you've been making them, because that's when the company was formed, Microbe TV Inc. We had an older company, Microbe TV LLC, which is not a nonprofit, but Microbe TV Inc. is our parent company. And if you'd like to contribute to us, go to microbe.tv/contribute. You could do PayPal, you could do Patreon. I have a Venmo. I didn't put the link yet in the show notes, but I have to do that at microbe.tv/contribute. Or you could send a check if you want. Send me an email, Vincent@microbe.tv, and I'll give you an address to send the check. Having trouble getting mail at the incubator. So I'll have to give you another address because the post office thinks we don't exist. I don't know. I think I have to go into the local post office and fix it, but I haven't had a chance. I'm just blown away by the fact that humans have advanced to the level that we can do individual sequence. Yeah, it's very, you can do your individual genome. You can do what's in your respiratory tract, your gut tract. It's great. But as Amy would say, we don't know what to do with a lot of that information, right? Have I heard of folks with long COVID being treated with monoclonals and alleviating their symptoms? I haven't heard that. Daniel would have mentioned it. No, I haven't heard that. So what about herd immunity? So herd immunity is when you have enough of the population immune to protect the people that are not immunized, and that fraction depends on the virus. I don't know what it is. It was depending on the original R naught of SARS-CoV-2, it was 70%, but that may have changed. However, the point is that vaccination drops transmission. There's no question. It drops shedding of infectious virus. It's going to drop transmission, so it's going to impair transmission in the general population. So the likelihood that an unvaccinated person will get infection drops. It does go to zero, but it drops, and eventually the epidemic fizzles out because there is not enough virus to infect everyone. So when does this happen? Well, so herd immunity is another difficult metric, because if you take the world population and you average it, we're not at 50% globally. So, but pockets can be 90% and pockets can be 30%. And so you can always have pockets of people who are not very immune, and then you can have outbreaks there. So you have to think about herd immunity as not just on a global level, but on a local level. And you can have an isolated town that has 30% immunity, and if the virus is introduced by a traveler, you'll have an outbreak, despite the fact that the rest of the country has so-called herd immunity. The Great Barrington Declaration was nonsense. It had no scientific basis whatsoever. It was a political ploy, and we dismissed that very readily on TWIV, in fact. Former smoker of 20 years gotten 2 Jansens. I think if when you stop smoking, your lungs after time recover. So I do not think that you are more susceptible as a consequence. If Omicron can displace Delta despite losing its furin cleavage site, is this a warning that sarbecoviruses have other paths through which they can become pandemic capable? Absolutely. And it didn't have to go through the ancestral virus, right? Sure. Many ways to infect people. I'd be on campus after Martin Luther King Day. So the first 2 weeks at Columbia are remote. So I will be on campus after those first 2 weeks, which I think brings us to the beginning of February. Then I'll be on campus. I'll be in Havemeyer on Mondays and Wednesdays, 4:10 p.m. Yep. And the course at Columbia is not live. Well, it's in front of a class after 2 weeks, yes. But I'll record all the lectures and post them as usual, just so that you can see if anything changes. Wow, we're not even halfway through tonight. Let's see if we can rip through here and get to some people who haven't had a question here. And mice and deer, in fact, we have to monitor them. We have to do more surveillance of mice and deer to see what's in them and the extent of infection and so forth, to see if it's a threat. Sure, absolutely. What should happen to the cruise industry now? I don't know. If they want to require vaccination, that would probably cut down on the issues. People wouldn't get sick on board, right? But if you have an unvaccinated person, they're going to get infected for sure. Unless you do testing beforehand and make sure no positive people come on board. Do we have nicknames? No nicknames. I call her Amy and she calls me Vincent. And never mind. Oh, moving on the halfway point. Thank you, James, for your contribution. We have developed a face mask that eliminates virus using copper circuitry. Cool. We can't do it. We don't have a lab at the incubator, but you can certainly find people who would do that with you. Oh, boy. If any of you are billionaires, please fund Amy and Vincent for life so she has time to talk to the press and straighten them out. Well, she would like to be funded so she can continue to do her work. Right now the funding situation is in the U.S. is very tough. We do need some private support of research from people who can do it. I do think we should explore that more than it has been. Oh, and yes, Steph, one of our moderators, says I can answer any deleted message. I see them all in my streaming software. So there are 2 ways I look at it. And this way, I see all the comments whether they've deleted them or not. So, but I usually can tell which ones don't need to be answered. And we're just trying to keep it clean. They do a great job. Hello, Vincent and Amy. It's a tradition for me and my 7-year-old daughter to watch your live stream Wednesday nights before bed. How would you explain to a 7-year-old the way vaccines work? Vaccines, protect you by being like a virus, but not doing any of the nasty things that viruses do to you. So I don't know what a good analogy for that would be, but we've changed the virus enough so it doesn't hurt you, but it helps you. And good for you for listening. Good night. Can they study HPV vaccine? Sorry, that sometimes when I click on a comment, it highlights someone else's to understand why it's sterilizing. They could. They know that antibody levels in the cervical mucosa, the urogenital mucosa, I should say, are quite high, and they're maintained at high levels for a long time. And why that should be is not clear. It is a virus-like particle vaccine, and many people feel that the repeated nature of the particle somehow makes the immune response better. But I agree. I think it should be sorted out so that we could then apply that to other vaccines. Yeah. My daughter just recovered from COVID. When should she get her? Wait 30 to 60 days, I think. But make sure she's recovered before getting a booster. And good luck at Columbia. And if she gets in, she needs to take my course, of course. You'd love my dad. He was a microbiologist for the Air Force and Civil Service for 40 years. Back in the 60s, when those mad scientists, the Air Force guys, used to try vaccines on themselves, he's had over 50 of them. Wow. Cool. Do the data suggest long-term efficacy against severe disease after 1 or 2 doses? Well, I think some of the predictions have been 5 years, but these, respiratory viruses don't induce long-term immunity. I'm talking about 30, 40 years, like, say, polio and measles virus do, that have a blood phase. So I think even the T cell memory and the B cell memory is not going to last forever. But how long? Yes, we don't know yet for sure. I don't think Amy's arrogant, frankly. But if you want to make an excuse, and that's the kind of comment we don't want, calling Amy arrogant. Just go. Just go. Any hypothesis as to why people are getting tinnitus from the vaccines? I think it's neuroinflammation, yeah. Cytokine responses, inflammatory responses caused by the vaccine, yeah. Marty Macari said gain of function should not be done. Well, Marty doesn't understand that pretty much all of biology is gain of function experiments. So you want to stop biology? Fine. Any new property you give to any organism is a gain of function. So he thinks it's only bad stuff, but he's wrong because he doesn't know the nuances. And yeah, we have to do gain of function, and no gain of function ever led to a bad thing. So what's the problem? It's garbage. Garbage. Hemp comp. Oh, here's the hemp again. We have to see the data. This is a press release. No good. No good. Have they had any luck finding the mice that no, that Omicron? It's a hypothesis that Omicron evolved in mice, and that they would have to find it in mice, but nobody's looking very hard in mice. I have COVID, and people are telling me I should have boosted even though I'm fully vaccinated. Can you elaborate on this? Well, I'm assuming you're having mild symptoms, right? So you were twice vaccinated, probably 3 to 4 week interval. Now you're infected, so you're good. You don't have to get boosted. Infected with two doses is absolutely fine. Mark, you got questions from other people. I see. It's not just you, right? Okay. Let's see if we can rip through here. Do you enjoy having an audience that includes regular people? or you're hoping for a more credentialed audience. No, absolutely not. I don't want a credentialed. I'm sure you're credentialed in your own way, right? I don't want to talk to scientists. No. I talked to them all my career. I want to teach non-scientists about viruses. So I love that, we have 998 people here who want to talk about viruses, mostly. 99% of you want to do that. My talking to my colleagues is a different conversation, right? But I'm not terribly interested in that because it doesn't help set the world straight. So no, I'm happy. I'm really happy to do this. Thank you, Justin, for your contribution. How accurate are the at-home rapids? Well, the PCRs are quite accurate, but too sensitive. In the at-home antigen tests, if you are infected, they will be positive. They have a good track of being positive when you are positive. But if they are negative, I think in an antigen test, unless it's a false negative, so there's a certain rate of negativity, and it depends on the antigen test. There's so many of them. Each one is different, and the FDA has a website where they list them all in the, false positivity and false negativity rates. If you are negative, you are not infected, for sure, unless it's a false negative, which, as I say, happens at a certain rate. How quickly does the level of protection against severe disease conferred by the vaccines fade? Well, we haven't seen it fade yet. It's been almost 2 years right after, I guess just over a year after the first vaccines. So that's a good question. How long is it going to last? 2 years, 4 or 5? We'll see. And maybe that's why they have variant vaccines in the sidelines ready to go in case the protection against severe disease wanes. But so far it has not. And we don't know how long it's going to last. It's a good question. Yeah. Is it known why synonymous mutations can change phenotype? So a synonymous mutation gives you the same amino acid, right? And so RNA has structure that can be affected by these synonymous mutations. And so that could change the phenotype without changing the protein. We know many examples in viruses of changes of mutations in non-coding regions. There's no protein there at all. And they affect the structure of the RNA, and that can have a phenotype. I can imagine that the immune system searching 6 months for better antibodies leads to more accurate results, but do we know how somatic hyper is able to produce antibodies? Yeah, so when you get an antigen, a virus in you, your lymph nodes develop germinal centers, and there the somatic hypermutation occurs. And you make mutated antibodies, basically, mutated antibody genes and altered proteins, and they're selected for better affinity to the antigen. But the antigen doesn't stick around forever, so at some point that stops, right? So there's a certain month period of evolution. So if you get a boost, you can go through that phase again and broaden the response. That's the idea. With you going to the incubator to Columbia, taking the train, dining out for lunch, have you evaded infection? I have, apparently, but I have not tested myself. The last time I got tested was at Columbia. They do random tests by PCR, and I think it was 2 or 3 months ago, and I've always been negative, except for a very first false positive very early in the outbreak. I have no symptoms, but I haven't done any at-home tests. And yeah, I'm on the subway. I'm on New Jersey Transit. I'm around a lot of people, but I wear a face mask, and I haven't had any issues. No. Viruses are weird. Either they aren't producing enough to transmit, or the vaccine protected you. They are a little weird because they're not human, right? They have a different modus, and we can't really get it fully. And it's not just a matter of them going into everybody and infecting and moving on. They're weird. All right, so Paul, there's a suggestion that cell entry can be more efficient by endocytosis in cells and culture. It's not clear at all if that's happening in people. But yes, a single cell could be infected with both, absolutely, if the person were infected. But not in many places, there's only Omicron circulating, so it's less likely. that's going to happen. And Elise says, I think R is a useful concept in a naive population not taking unusual precautions. That's correct, but there's no such thing as R naught since it is always place and time dependent, and that's absolutely right. You have to realize the vagaries, and for someone to come out 2 weeks after Omicron is discovered and say its R naught is 13, it's just nonsense, because you can't do R naught there anyway, and you don't know the contribution of the virus to the and the population, the human behavior to it. You can't suss that out at that point. Vanity met Mikhail Gorbachev on a flight once. He reminded me of Mr. Rogers. I never met anyone famous. Did I? I ran into Woody Allen once on the street. I saw Anthony Hopkins once. No, and you think in New York, but I'm hanging out at Columbia. Oh, I saw the guy who wrote Salman Rushdie. Yeah, so I was leaving my lecture at Columbia, and he just got out of a cab on Broadway. I guess he was going to teach a class. Yeah, I guess he's famous, right? Yeah. Okay, let's start to wrap here. DNA Lady, thank you for your support. And I'm glad you think we have a message of sanity. If you've vaccinated and recovered from Omicron, is there any reason for you to mask up? I say no, unless you're required to. It's your choice, and I don't think you need to. So if you can go somewhere without a mask, it's fine. But if I go to a restaurant in New York, I don't need to wear a mask inside. And I've done that multiple times now in the last 3 months. I think you can walk around the street without a mask, but on the train and subway, I have to wear a mask, so I do. So no, if you're triple vaccinated or infected vaccinated, if you're not required to wear a mask, no, there's no need to. This is part of my philosophy. We need to move here, get going, back to life. We have vaccines that work. Reverend Sasquatch, hello. I didn't do any thumping tonight. Shirts that aren't buttoned down are useless. The only shirts I own that aren't buttoned down are T-shirts. Yeah, I don't like the non-button down. I just like this little anchor here, and I've always done it. You would like to take a virology class? Well, you could take mine online, but it's over. If you're in New York, you could get in. Nobody checks. If you want, ask me where it is. I'll tell you where it is. Did you hear about the Yankee Candle Amazon review method? Apparently reviewers say my candle came, but there's no scent increase during surges. Yeah, I love these folk tales. It's great. It's great. Thank you, Julian, for your contribution. Can COVID transmit in a public sauna? No, I bet not. Nobody's tested it, right? But that temperature, no way. Nope, not happening. There's a hemorrhagic viral outbreak going on in China. Why do you? No, I don't know that there's any evidence that there's any recombination going on. Let's take a look. Let's look at ProMedMail. ProMedMail, and let's see what's going on here. African swine fever, canine influenza, African swine fever, Middleburg virus. It doesn't even show up on the latest outbreaks here. Oh, it's so dominated by other things. So I don't know anything about it, but I doubt it's anything unusual, right? Thanks, Ian, for your contribution. Hello from Sydney. I'm about to get a booster. 4 months as recommended. Do you think another would be required? No, I don't think so. Because this is going to fix the too short first and second dose timing. So you don't need another one. Now, if protection against severe disease starts to decline, which it could, then we might consider something else. I mean, I don't think the immunity, the memory is going to last forever. It's not going to last 20, 30 years. But I think by then most of the world will be infected, and therefore it's going to turn into a rather mild respiratory disease, maybe a common cold. But so no, you're not going to need a booster unless the situation changes. And I don't see any reason why it would. I don't see any indication of it changing. Couldn't we engineer a non-harmful contagious virus that would vaccinate the public to combat anti-vaxxers? No, you can't. I mean, we can make attenuated virus vaccines, right? And the poliovirus vaccine, in fact, you drink and it reproduces in your gut. You shed it and it can infect other people and confer immunity. But we don't do that unwillingly, right? You have to agree to take the vaccine. So it's not something that we can do to vaccinate people who don't want to get it. No. In War of the Worlds, the thing that killed the aliens at the end was their exposure to a foreign something. I don't know if it was it was probably a bacterium. They had zero immunity against. Yeah, but that wouldn't have killed them. But it's a little bit of a problematic scenario, I think, right? Why would they all die? I mean, the thing is, there weren't many of them. So, okay, they were spaced out. And so why would they all get it? Maybe they got it from people. I think it's an unlikely scenario. So the thing is, in any population, you always expect to find some people who are resistant, right? So if their population was too small, if there were just, I don't know how many of the monsters there were, or the machines, right? If there were 50, okay, there weren't enough mutants in there, right? What's Paul Offit's argument for not getting the booster? It is that the protection against severe disease has remained constant in the high 90s, no matter what variant is around. He said that on Twiv. Go back and listen to him. Oh, yes, the sides were flipped. So I used the new computer tonight because last week the other one was acting up. And so I had to reinstall the streaming software on, and obviously I didn't have Amy in the right place. Sorry. I don't think it matters. Thank you, Shannon, for your contribution. Really appreciate it. I think I answered it, Andrew. I try and go through these one at a time, and I skip some that are repetitious. I try and get them all, and that's why I never get through everything. Has the vaccines been too spike-centric? Yes. I think we should have had more epitopes, which would give you more T cells. And in fact, if there is a next generation vaccine, I think they should put other viral proteins in. But the idea was that the spike antibodies are going to block infection. So let's go with that. They didn't know anything about anything else. And that was based on experience with other coronaviruses. But I do think having other epitopes in there would have helped. And many people think so as well. Thank you, Chris, for your support. I really appreciate it. Thank you, H. Sue. Where do I find the address to spend something? Just send me an email. I think I have your email. Vincent@microbe.tv, and I will give you an address. Give you Columbia address. Oh, by the way, besides what we said earlier, as you know, Tony Fauci last week said he doesn't care about how many cases there are. He just cares about hospitalization. So it's another idea that we should worry less and less about just infected people and obviously the ones that are getting sick. No evidence of SARS-CoV-2 infection in the brain. 1/12/22. That's today. Yes, there is no evidence. Well, it's not my evidence. I'm just reading the literature, right? I watched Cells at Work. I have the new one. So two holidays ago, my son Devin gave me, oops, it's backwards. The original Cells at Work, the manga, which is great. You know, it's all about, she's a red blood cell there. You can see he's a white blood cell. It's great. And then he just, for this holiday, he gave me Code Black. Cells at Work, Code Black. So cool. I love it. This is kind of adult because there's a lot of blood in it, but this is other things besides what cells normally. It's really good. Okay. What is the appropriate dose interval for Pfizer dose following COVID infection? I don't think you need a booster. You had COVID 6 months after. Wait a month or two after the infection if you want a booster, but I don't think you need to have one. If T cells are mostly important to protect us, why bother with the third shot? Well, that's a good question. We didn't have the data. They're just pouring in now about whether 2 shots 3 to 4 weeks apart would protect you against severe disease against Omicron. We didn't know that. And it may be that, never mind. Yeah, so we don't know, and I decided to take the risk. But I understand if you don't buy it. I totally get it. Okay, so yes, you can find viral RNA in many sites in the body, but it's just by PCR. It's not an infectious virus. It probably is meaningless. It's just in every infection, no matter if it's a rhinovirus infection, you could probably find RNA elsewhere in your body. It's meaningless. So if natural infection and the vaccine are essentially the same mechanisms of training the immune cells, how are vaccines any different or better? Well, they're different because they don't kill you. Is that good? Vaccines are made not to kill you. If you want to get COVID, you have a good shot of being killed. And the rate of killing depends on how old you are, but it's not zero at any age. So if you want to take that risk, it's fine. It's your choice. I'm not going to argue with it. I think for polio, for example, a lot of kids got paralyzed permanently, and so the vaccine prevented it. Yep. Do I think vaccinated runners should be forced to wear masks while racing? No, absolutely not. As I said, vaccines are working. We should move on. Daniel Griffin has a chart of the progression of the disease. Has there been any reason to change or update? Well, see, he seems to be thinking that Omicron has accelerated the process, but I don't think those data are in yet. And I think we need to wait a bit. It could be revisable, yeah. In fact, I think last week he had some new charts, which he really liked. So check out his last update last week for that. Thank you, Doreen, for your contribution. Here tonight for my dose of science and logic. Hope you got it. The temperature is 2C. That's great. TWIV is a warm breeze. The TWIV will be dropping at midnight Eastern time. I edited it today. And it's not COVID. So, but I think it's really interesting. Atul, thank you for your contribution. And I know that if there's some of you who took the live stream virology course, I know I didn't put the exam up yet. I plan to do it. I always find myself running out of time. Like I should go now and do it, but I'm loving answering your question. I'm going to go to 10:30 again tonight. Yeah. Does the flu shot help versus COVID? No, it doesn't. I know there was some idea in the past that the flu shot might help. But, I mean, I see no evidence that it does. So that's what I should tell you. I shouldn't say it doesn't work, but it depends on which one you get. There was, the flu mist would be the one that might give you a couple of months protection. But if you're vaccinated for COVID, it doesn't matter, right? Have you seen the Japanese paper that says infectiveness of droplets declines quickly? I didn't see the paper yet, but I'm going to look at it because I saw the the Guardian headline today. And so that could be very interesting, right? A common argument of anti-vaxxers is that vaccinated transmit as much as unvaccinated. So this is wrong. It's based on that crappy CDC Provincetown report that I mentioned. And they don't. They don't. This is phony, and the press needs to counter it, but I'm not sure they can. Thank you, Ditching. Appreciate it. What contributes to the ability of a virus to create many variants? So it all has to do with the mutation rate, right? The ability of the virus to make lots of mutants. And it does so. So RNA viruses like coronaviruses and influenza viruses make a lot of mutants because they have no way to correct mutations that get introduced during copying of RNA. DNA viruses are less mutation prone because they can fix the mistakes. So as long as a virus is replicating in a cell, you'll get mutants. So if a virus comes in your nose and replicates in 10 cells, each of those cells will make viral mutants. The more cells, the more mutants. The more people infected, the more mutants you'll get. And then you got selection to pull out the ones that matter. So what kind of selection is there? Well, it could be the ability to spread from one person to another. Fitness, that's part of fitness. It could be reproduce faster, reproduce to higher levels. We don't know what the case is for any of the SARS-CoV-2 variants, but we're starting to figure it out. But it's a good question. I'm sorry. I'm looking at the next one. Did you see Lex Friedman's Jay Bhattacharya? I don't know. He might be my Columbia colleague, if he's who I'm thinking of. I don't think we need lockdowns anymore. No. I think, I talked to a Danish reporter the other day, and he said in Denmark, where they have over 80% vaccination, they're thinking of locking down. I think it's absurd. Don't. If, you're infected, you're not symptomatic, there's no reason. Just go forward. You can, and the only issue are kids, right, who are not vaccinated yet, who are older people. No, you don't need to lock down. What day will you be discussing the new paper? The one I mentioned, the viral load, I'm going to do Friday. I'm a little disappointed in Lex. He had me on to get the truth, and then he's had a whole bunch of people who spew nonsense. But it's his show. He wants to make the bucks, and he's making a lot of bucks. It's his show. What the heck? Thank you, Westfield 90. Appreciate it. After so many Omicron infections, is it time to start recognizing natural immunity, at least as a passport? The problem is, what can you measure? You get 2 shots or 3 shots, you can put that on a card. So what are you going to do? Show a positive PCR test or a positive antigen test? Is it the same? So that has to be worked out before that's sorted out. I think people are thinking about it, but I think it's a fair question. So I'm working on a show with Lori Garrett, just to let you know. I'm going to have her do a show in the Microbe TV network. We'll see if she wants to do it. I think it could be fun on public health. Talked to her last week about it. The Atlantic is good. Karen Shu, I think her name is. Great writer. The T cells will save us, her last article. She gets it. Okay, we need to get close to the end. Here we go. Marge, thank you so much for your contribution. You guys are great. I can't do the incubator stuff without your contributions. So thank you so much. And I did talk a bit about Corbevax, where I was worried about them using antibody as a surrogate for protection. Thank you, Paul, for your contribution. PCR measures the presence of viral RNA, doesn't indicate infectivity, but if one had a positive PCR It would change. I don't know what you mean there. If you have a positive PCR, you might be at the end of an infection. If you don't feel sick, what's the difference? That's our point, I think. Thank you, Peter, for your contribution. You're welcome. Should I wait to get boosted if there's an Omicron updated booster? Not going to be one anytime soon. So don't wait. Don't wait. What do I think of the Yorkovich? I don't know what to think of it. I think they kind of bungled it down there under, right? Just keep the guy holed up without... I don't know. It seems not fair. I don't know what the latest is. This is a guy who won the Open, 9 times or something, and put him in a hotel. I mean, I know he's not vaccinated, but didn't he... I don't know. He got an exception or something. I don't know. I don't know what to think about it. It's, in the broader issue of, a sports person and what to do. Does he have a valid excuse for not vaccinating? I just don't know the issues. I'm sorry that I can't be more useful, but I'm more useful with viruses. Thank you, Roadhouse Mike, for your support. Online virology is there, yeah, and you can see all the questions people asked. It was fun. I really miss it. I'd love to do it again, but I have to do other courses, so it won't be. And in the fall, I will teach a virus course. It'll be called Viruses Live. It'll be each lecture about a different virus. Okay, so come for that. It should be fun. Thank you, Stuart, for your contribution. And I can imagine doing several lectures on herpes and coronas and so forth, and going into the details about all sorts of things. Thank you, John, for your contribution. Really appreciate it. Thank you, Ian, for your contribution. As always, appreciate it. Thank you, Justin. You guys are great. I'm really amazed that, someone... DL Sendero, you sent me an email, so now I know who you are. Thank you very much. At the beginning of the pandemic, someone said, why don't you... we started these live streams, and someone said, why don't you turn on the super chat? And I didn't do it, because I said, what the heck, But, and at the beginning, people didn't like it, because I tend to thank people, right, and they don't like that. But it's been great. It really has raised a substantial amount of money to pay for the incubators. So thank you, Magpie B, for your support. You don't have to support us to ask a question. I've answered a lot of questions for people who didn't donate, but I do want to thank people who donate. Do you require a vaccine passport to visit the incubator? I think you should be vaccinated, but I'm not going to check your passport. The incubator is near Italy. Oh my gosh, what a great place. If you come to the incubator, you have to go to Italy for lunch. There was something here I wanted to... I think I talked about boosters quite a bit. These questions are probably old. I think we explained them, how they work. As a doctor here in New Jersey, I learned more virology with your book than when I was in med school. Well, thank you. Thank you very much. Can principles of virology be picked up at the incubator? I don't have any copies there. I might at some point. If you come, just let me know, and I could have one ready for you. Why haven't we seen more emphasis on intranasal vaccines? They're in the works, as far as I understand. They are in testing, and, it'll be a while. And testing a vaccine now that we have good vaccines that work is harder. So it's going to take longer for them to get out there. Great questions here. I'm sorry to pass them all up, but now it's time to shut down. I don't want to keep the mods any longer. Simon, thank you for your contribution. Thank you, Todd, for your contribution. Appreciate it. I want to thank all the moderators tonight. Vanity Nutrition, Les, Tom, Frank. I think I got them all. John, thank you so much for your contribution. Overjoyed, there you are. I haven't seen you until now. You've been quiet. You met David Baltimore and everyone else in the field. Well, that's true. So David Baltimore, I postdoced with, right? But I've met a lot of people in the field because I go to meetings and I interact with people. And now that I've been podcasting for 13 years, I've interviewed a lot of them. Yeah. I think I've interviewed more virologists than anybody else, even through this pandemic. What makes a virologist? Curiosity and passion. Oh, yeah, you need to do a PhD in virology. Well, you don't need a PhD. You could have a master's degree or a bachelor's and work in a virology lab, and you'd be a virologist. But you have to do experiments and do research. Well, I guess you could do quality control in a company with viruses. You could be a virologist. So there are a lot of things you could do. You could do production of vaccines, and you'd be a virologist. You don't have to do research. So there are a lot of different paths you can take to becoming a virologist, not just the one that I did. But I do think that passion and curiosity is essential. And if you've ever been bored, you should not become a virologist or any kind of scientist, because there's no reason to ever be bored in this world. If you're bored, it means you're not paying attention, because I've never been bored in ages. I can't remember the last time I was bored. I was young and I didn't get it. Thank you, Lisa, for your contribution. And don't someone tell me I'm being arrogant. Once someone told me last week I was arrogant. I'm not arrogant. If I'm not bored. I'm just fascinated, and I could stand in line and not look at my cell phone and look at things or think about things. I take the train every day, and I'm always fascinated by what's out the window. I can always find something interesting, but I'm usually working on my laptop. Thank you, Michael, for your contribution. Really appreciate it. Thank you, Jerry, for your contribution. Thank you, A. We'll keep making TWIVs as long as I'm around. Well, my goal is to make a company that goes beyond me. I'm not going to be around forever. And hopefully I can do that and get other people interested who can bring it forward. And so what is the requirement? Passion and curiosity. Yep. Clinical virology. Yes. Yes, of course. There are many types of virology out there. So that is what you need. Okay, folks, you are great tonight. And I appreciate your coming here. Thanks to the mods. Thanks to your great questions. And yes, if you visit the incubator, we can bring eggplant parmesan. I have to get a microwave, I guess, but got a kitchen in the incubator. Come back next week. I'll be here with Amy, and bring your questions, and we'll answer them to the best of our ability. Meanwhile, listen to TWIV, 2-3 episodes a week. Tomorrow we have a TWIP in person. A lot of cool science podcasting happening at the incubator. Thanks, everybody. Be safe. Have a great week. Thanks for coming. See you next time. Good night. Oh, I don't have my bloody microbe TV thing here. Let me bring it up. There you go.